Hey, my name is Jamarian Williams, a freshman chemical engineering student at the illustrious North Carolina Agricultural and Technical State University, and this is my biology project. So the protein that I have right here is a type 1 collagen, and the mutation of that would be a dominant negative substitution of glycine for a larger amino acid in several locations. It can also cause brittle bone disease, which is called ogenesis imperfecta, which I have right here. And then you can see the loss of a triple helix right here. And then the type 1 collagen is a structural protein shaped like a rope in a triple helix structure. It prevents tissues from stretching and tearing and provides strength and support to various body tissues, such as bones, skin, tendons, ligaments, and the cornea. So right here, this is a mature type 1 collagen, basically what I just explained. And then this is the mutant type and the wild type, basically the normal form of it. I chose the structural category because this is the main function of a type 1 collagen. It is to act as a scaffolding, helping maintain the integrity and shape of connective tissues throughout the body. The subcellular location is synthesized in an endoplasmic rectum, modified in the geo apparatus, and then secreted outside the cell into an extracellular matrix. Cells that typically contain type 1 collagens are phorblasts, osteoblasts, bone cells, and sonodontocytes called cartilage cells. It is also found in organisms like animals and humans, especially vertebrates. The protein becomes less active because the substitution of glycine, a small amino acid, for a larger one disrupts collagen's triple helix structure. This makes the fibers weaker and more prone to breaking, leading, and brittle bones, other symptoms of osteoarthritis imperfecta. So right here, when I was talking about a bigger amino acid, this is basically when it goes to a wild type to a bigger mutant type, where you can see a big white amino acid that messes up the type 1 collagen. And this could cause diseases like this osteogenesis imperfecta, and then a loss of a triple helix basically happens in that. And it becomes bigger, so you can't really fit it where it needs to be fit. So a glycine has a hydroatom, and as its side chain, which is the smallest possible group, and its function group is a negative H nonpolar. So it is slightly nonpolar. It is neutrally charged, and it is the smallest out of all 20 amino acids. So right here we have the collagen alpha helix amino acids, and then we have all three right here. The amino acid sequence, we got glycine, proline, and then hydroproline. And then glycine is so small that it also allows tight packing in the collagen helix. It does not form strong bonds, but is essential to maintain its shape. The structure of the mutated side chain is a negative CH2, negative OH. This negative OH makes the side chain polar and capable of hydrogen bonding. So the hydrophilic is polar, it's a neutrally charged, and it's larger than glycine. Glycine's small size allows the collagen strands to twist tightly. Serine or other larger residues cannot fit in those tight spots and bulge out, disrupting the helix. This leads to fragile collagen, which causes structural problems at the cellular level, and organism-level bones break easily. The protein cannot form strong collagen triple helixes. The cell also produces a defective extracurricular matrix, and the organism's function leads to brittle bones, frequent fractures, and development problems in tissues requiring the collagen. This is the mutation that causes all of this. The scale of the model, I'm talking about limitations of my model, is the scale of a type 1 collagen. As I said, it's really small, so this is not the same scale of it. And then on top of that, another limitation would be the motion. And my model is static, but in real life, the real proteins are dynamic and flexible. And the 3D model is also simplified. Complex interactions like hydrogen bonding and hydroxygen of collagen are not shown in detail. So also, I want to show all the mutations, and basically my full project. I don't think I really explained it all the way, but it's a type 1 collagen. We have the mature type 1 collagen right here, AM1, and then we have the wild type and the mutant type. And you basically see the bigger amino acid. And then we have the disease that it can cause if it doesn't work properly or a mutation happens where you lose a triple helix, and that's how you would get that disease. And then also we have our alpha triple helix amino acids. We have the amino acid sequence, and that's pretty much it. Thank you for listening to my video, and have a great day.